Now we're going to talk about what happens once transformation has taken place. So great, we have gotten to the point where we have a plate full of E. coli bacteria that have successfully produced the protein that we care about. In the real world, it would be insulin. In our example, it's green fluorescent protein. Perfect. But we need to get to a point of actually isolating just that protein away from all of the others that are in the bacteria, because it's not like we just scoop up a bunch of E. coli and give it to a person with diabetes and say, here's your insulin, here you go. So enter protein purification. There are a couple different ways that we can go about doing this, but again, the idea is that we purify the one particular protein that we care about away from all of the others that something like a bacteria would produce, and all the other things that aren't protein in a bacteria. Like, we don't need the cell membrane and all the other organelles and all that stuff, just the one protein. So this process is called chromatography. And for us, there are many different kinds of chromatography. It's just a big fancy word that means a separation of elements of a mixture. So if you think about having a mixture of things, and if I said, organize it, you could do it in a number of different ways. There are size or shape or specific properties. The one that we're going to focus on in our experiment is called size exclusion chromatography, or SEC. This basically separates proteins by size. And since, we know the exact size of green fluorescent protein because we know the sequence. So if you know the sequence of something, you know how many letters it is. Therefore, you know how many amino acids it is, how much size it should, in theory, take up. You can use that information to kind of select just for that particular size. So how do we go about doing this? Well, we need to grow tons and tons of bacteria. So basically use them as tiny little factories to make as much of the protein as we need. Then we destroy the bacteria. So we need to essentially lyse or explode the bacteria to get to the proteins. And then we separate it using a column, kind of like what's seen on the right here, to separate out those elements of a mixture. In our case, it's a little bit easier too, because green fluorescent protein glows in the dark. So you can actually see it as it moves through. Not all proteins you can do that for. So this is kind of a rough picture on the right of how size exclusion chromatography works. Since we know the size of GFP, we know exactly how we might want to filter it in order to isolate the GFP. But again, as I mentioned, you can see it. So you can kind of watch it with a UV light as it moves through the column. The column itself is filled with a beaded matrix. And the matrix of different sizes can separate all the proteins. If you look at the picture, depending on, what happens, you dump all of your sample here at the top. So you would dump all of your exploded cells here at the top, and they would slowly begin to migrate through this matrix of all of these different beads. If you have a small molecule, like a small protein, then it might enter the pores in these tiny beads. So think of each one of these beads as like a tiny wiffle ball. They have holes. So if you have a small protein, that small protein can go through those little holes and kind of get stuck. And then it takes them a while, but they work their way out. So small molecules end up getting stuck in these wiffle ball-like beads longer than the medium or larger size. The medium molecules kind of get stuck sometimes, and other times they don't. So they end up kind of moving through at a medium pace. And the big fat molecules can't fit through those wiffle ball-like holes. So they just move right on past those beads, and then they can travel rapidly. So the idea is if you add it all up here at the top, based on the size of your protein, it's going to make its way through at a different speed, where the big fat molecules will end up coming out first, then the medium, and then the small. So if you know what size your protein is, you can kind of gauge when it should be coming out. And then you basically put your little tube underneath and collect the fraction or the piece that you actually want. So that's the underlying concept of size exclusion chromatography. And this slide just kind of goes through that same element that I just talked about. Smaller things are going to get trapped in the beads a little bit more, so they're the ones that are going to end up coming out last. And here's how the process works. So we would take our, solution of exploded bacteria. So we would have harvested the bacteria. We would have treated it with a lysis solution, exploded the cells, and now the GFP protein and all the other stuff is just this mush mix. So we add it to the top, and we unplug the bottom, and it starts to drain. Then we kind of add some different buffers along the way, getting the proteins to move their way through the matrix. Again, smaller ones are going to come out last. The big fat ones come out first. Because it's fluorescent protein, we can actually watch as it slowly moves through. And then the idea is when your protein is getting close to the bottom, you start to purposefully collect it. So instead of just having it drip into a waste container, you're starting to actually purposefully collect it. We can use tiny wells that collect different drops or different chunks of the liquid, but the idea is you do it until your GFP has come out, and then you cap it back up and are done. When you look at it with a UV light, you can actually see maybe one of the wells is super glowing compared to the others. And that means that maybe then you've actually successfully isolated GFP all by itself. So that tube would, in theory, be the pure harvested protein that maybe then you would give to an individual. So if it had been insulin, maybe that's the tube that contains just insulin and nothing else. But it is possible that you might have captured some tag-along proteins of similar size. So if that happens, then you need to do the final step, which is verification that you did indeed get what you wanted. And that'll be in the next short video.